Yes, good evening to all. My voice is audible. Please let me know. My voice is audible to you. Please let me know. Hello, my voice is audible to you. Please. Okay. So, good evening, Diksha. How are you? How are you, Diksha? How are you, Diksha? So, continue. हाँ 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 ठीक है। चलो दोस्ती आ गई है हम से में। उसको एक मैसेज भी कर देना कि मंडे से क्योंकि उसकी टीम्स पे ऐप नहीं ना पर बनी हुई। हाँ कि अपनी अपनी टीम्स पे ऐप बनाओ कि हम ऑनलाइन क्लासेस ले रहे हैं। ठीक है ठीक है। So yes, today we have to start another lecture which is there in the previous class. So what we have done yesterday, Diksha, what we have done yesterday? The topic which we completed yesterday. Yeah. So please tell me so that I will continue with that only. So we have covered these hormones, we have covered enzymes, lipids are also covered, fats are also. Lipids we have done, fatty acids are completed, amino is okay. I have to continue with the protein part. I think so. The protein part is the one which is left, right? So we have to continue with the protein part because we have just yesterday we covered amino acids and now we have to cover the protein part. So I also gave you homework regarding the amino acids also. I hope you all have done it, but I didn't get any message in the group also. Rather, none of you have pinned it also. So I request you to complete your homework as well as please pin the question so that all will able to reply or all will able to answer the question. So we have to cover the protein part today, which is left yesterday, right? So we have covered amino acid yesterday. Now we have to complete the protein part. So what is the homework yesterday? Please let me know what is the homework which I gave to all of you. Please let me know. So have you completed the homework? What is the homework yesterday? Please answer me, please. Why your students are not attending the classes? Only three or only two students are right now present along with us. Why they are not attending the classes? They don't want the certificate. They not want the certificate for which they pay Why they are not attending the classes right now? Otherwise, it will become problem for all of them because your attendance is counting. So I request all of you, why you are not attending the classes? Only 5 students are present right now. Only 4 are. Okay. So I request all of you. Please reply all of you. Have you done the homework? I want the reply from all of you. You are not attending the classes also, and please, none of you have completed yesterday's homework also. So please reply. So I just want to know you one thing that okay, you are not taking the classes, it's okay, but assignments is compulsory for all of you. The homework which I gave you, you have to complete it. Otherwise, you will not get your certificate. Okay, so please try to send me your assignments on time. Please try to send me as soon as possible. Are you getting me or not? Please let me know. Okay, okay. So because of some exams, some students are not able to send me their assignments. It's okay, but you are not present in the class, and uh, I am just talking about all the students. There are 40 plus students in this biochemistry class, but none of them are present continuously or regularly, and moreover, your assignments are also not on time. So I will not repeat these things again and again. It will later on. It will bother you during your certificates. So this is my last time I told you all that your assignment should be on time, right? And also, if you are not be present in the class, I will complete the topic. You When you will be able, you can watch the lecture. There is no issues regarding the lecture, but at least try to be complete the assignment. Okay, so I know you have not completed your yesterday's assignment. Complete it and send it in the group as soon as possible. Get it? And I want the reply from all of you. Please, it's my request to all of you to reply, so that we will start the session. And what is the scenario of your review paper now? How much you have done the review paper, or how many of you have completed it, the abstract and the topic? Please share with me so that I am able to know. Please share. Yes, please share with me the status of your abstract, please. Okay, okay. So the abstract is completed. Okay. Diksha has not submitted the abstract. Why? Okay, due to the practice assignment of college. No worries, but try to submit it. No worries, but try to submit it. Okay. Ayushi has abstract me the mail. Okay, thank you, Ayushi. Thank you for it. I will check it on this weekend by Saturday and Sunday. I will reply to all of you. Okay, so no worries. I will reply because only weekends I will be free. So I will reply to you by this Saturday or Sunday. Okay. Please try to submit it as soon as possible. Okay, Ayushi. I will check your abstract also. So shall we start now? So I'm not now just going to start the protein part because only I have covered the amino acids yesterday. Now only the protein part is there which is left. So I'm going to share it with all of you. So this is amino acid which we have completed. So amino acids are a group of organic compounds. We have already completed it yesterday. Then we have types of amino acids. We have standard amino acids. We have non-standard amino acids. After it, we have the amino acids on the basis of the like essential amino acids, non-essential amino acids. Then the amino acids are classified on the basis of their 20 amino acids. They are classified on the basis of their protein structure. So they are classified on the basis of the structure. And we have this is your homework also. I suppose all of you have to send me this assignment as soon as. Then after it we have the amino acids which are there on the basis of like aromatic amino acids which have a phenyl which have a ring. Then after it we have the classification of the amino acids on the basis of polarity. So we classify the amino acids on the basis of polarity. After it, so this is the polar amino acid with no charge on R group. This is the polar amino acid with positive R group. Polar amino acid with negative R group. So then the classification is on the basis of nutritional value. So according to it, we again classify the amino acid. Okay. So we have 10 essential amino acids which are there, and then we have some non-essential amino acids also. So based on their metabolic phase, we have 10 essential, non-essential amino acids also. Then we have glycogenic amino acids, ketogenic amino acids, glycogenic and ketogenic amino acids. So these are the amino acids which we have done yesterday. After it we have this GABA protein also. And then we have some properties of amino acids. We have some physical properties like solubility, melting point, then taste is there, optical properties is there, then amino acid and ampholyte is there. Zwitter ion is a term which we have discussed, which we have studied. Zwitter ion, or you can say dipolar ion. Zwitter ion is a hybrid molecule which contains both positive as well as a negative ionic group. After the Zwitter ion, we have some chemical properties of amino acids like the color reaction which is there, then transamination is there, oxidative deamination is there. After it we have completed some non-amino acids also. Then we have to focus on the protein and peptides. So peptide formation is there by dipeptide bonds. So we have a peptide bond. We have some functions of peptide bond also. Yeah. So we have also studied the basic structure of protein. So from here we have to start the protein part basically. So the structure of protein. So protein have different level of organization. We have different structures. We have a primary structure, secondary structure, and tertiary structure. So primary structure is basically the linear structure. So primary structure is the linear sequence of amino acids forming the backbone of the proteins. Secondary structure, the spatial arrangement of protein by twisting of the polypeptide chain is the secondary structure, and tertiary structure is the three-dimensional structure of a functional protein. Okay, so we have four types of protein structure which are there: the primary, the secondary, the tertiary, and the last is the quaternary structure. So this is the primary structure, which is the simple one. So this is the primary structure. After it we have the secondary structure, which may be alpha helix or beta plated. Then the tertiary structure is there. The quaternary structure is little bit more complex because it consists of two or more polypeptide chains which are referred to as subunits. So we have two or more subunits. The spatial arrangement of these subunits is known as the quaternary structure. So this four subunit structure is known as the quaternary structure. Now the structure of the proteins, the primary structure, the linear sequence of amino acids held together by the peptide bonds in its peptide chain. The peptide bonds form the backbone. The free NH2 group of the terminal amino acid is known as N-terminal end, and the free COH is known as the C-terminal end. So we have the N-terminal end in which we have the amino group attached, and we have the C-terminal end in which we have the carboxyl group attached. So it is a tradition to number of the amino acids from N-terminal end and as a number 1 towards the C-terminal end. So presence of specific amino acids at a specific number is very significant for a particular function of a protein. Getting my point? So a presence of specific amino acids at a specific number. So we have a proper amino acids list with a specific number. They are very significant for a particular function of a protein. So this is the primary structure of protein. Now we have determination of the primary structure. So the primary structure comprises the identification of amino acids with regard to their quality, quantity, and sequence in the protein structure. So we have to keep in mind the quality, quantity, and the sequence. Now this is a determination of primary structure. So the determination of primary structure basically involves three stages. The number one is determination of amino acid composition, degradation of protein or polypeptide, determination of the amino acid sequence. So this is the determination of amino acid composition. The protein or the polypeptide is completely hydrolyzed to liberate the amino acids. Now what is there? Basically when protein or the polypeptide chain is hydrolyzed, when they are break down, it will lead into the amino acids. The hydrolysis may be carried out either by acid or alkali treatment. The hydrolysis may be carried out either by acid or alkali treatment or by enzyme hydrolysis. So by hydrolysis, basically what we will get the amino acids. So pronase is a mixture of non-specific proteolytic enzymes that cause complete hydrolysis of the protein. So we have the enzyme pronase, which helps in the hydrolysis of the protein, which will lead us to the amino acids. So separation and estimation of amino acids. Now the mixture of amino acid liberated by the protein hydrolysis can be determined by the chromatographic techniques. So when we will get the amino acid by hydrolysis, now we have to separate them. For separation we need a chromatography. So chromatography is basically for the separation of the proteins or amino acids. Now after it we have a degradation of the protein or polypeptide into smaller fragments. The number one is hydrolysis. So by hydrolysis we will get the amino acids. The number two is degradation of the protein into smaller fragments. So liberation of polypeptide. The treatment with urea or guanidine hydrochloride. This disrupts the non-covalent bond and dissociates the protein into polypeptide. So by the use of some chemicals, we will able to break the protein into the polypeptide chain. So by using urea or guanidine hydrochloride, we will disturb the bonds and after it we will get peptides from the proteins. Okay, so cleaving the disulfide linkages between the polypeptide unit, treatment with sulfuric acid is also necessary. So we have to cleave the disulfide linkage. For it we need sulfuric acid. Like here we need pronase enzyme. Here the need is of sulfuric acid. Now number of polypeptides. The number of polypeptide chains which are there by the breakdown of proteins can be determined by the treatment of a protein densyl chloride. So it specifically binds with N-terminal amino acid to form densyl polypeptides which on hydrolysis yield N-terminal densyl amino acid. The number of densyl amino acid produced is equal to the number of polypeptide chains in a protein. So this is another enzyme, or you can say chemicals. It is densyl chloride, which is helpful in the breakdown of the proteins into the polypeptide. After the breakdown of polypeptide into fragments, now when we will get the polypeptides from these protein chains, now these polypeptides can be further breakdown into smaller peptides by the enzymatic or chemical methods. So we have enzymatic or chemical methods by which we will be able to break the polypeptides into smaller peptides. So enzymatic cleavage is the proteolytic enzymes such as trypsin, chymotrypsin, pepsin and elastase exhibit specificity in cleaving the peptide bonds. So we have some enzymes. Please remember these enzymes. We have trypsin, chymotrypsin, pepsin and elastase. They will break the polypeptide into the smaller fragments. So trypsin is the most commonly used enzyme for the breakdown of the protein into the polypeptides. So if you are able to see here, this is the peptide which we have get. So cleave with trypsin, determine the sequence of peptide using advanced method. So by the treatment of the trypsin, we will able to get the shorter fragments of the peptide. So these peptides are there. Then these peptides their sequence can be determined by the advanced method. Similarly, we have to cleave with the cyanogen bromide, determine the sequence, and after it we have to determine the sequence also. So trypsin hydrolysis is a peptide bond containing lysine or arginine on the carbonyl side of the peptide linkage. Cyanogen bromide is another important chemical which is basically used to split the polypeptides into smaller fragments. So from these polypeptides we will get the smaller fragments. So cyanogen bromide specifically splits peptide bonds, the carbonyl side of which is contributed by the amino acid methionine. So we have the amino acid methionine. Then after it we have determination of the amino acid sequences. There are some methods, there are some reagents which are there which will help us to know the structure. Like we have Sanger's reagents which will helpful to determine the insulin structure. So we have Sanger reagent has a compound which is 1-fluoro-2,4-dinitrobenzene. It specifically binds with the N-terminal amino acid to form DNP derivative of a peptide. This on hydrolysis yields DNP amino acids and free amino acids from the rest of the peptide chain. DNP amino acids can be identified by the chromatography. Then we have advanced reagent. It reacts with the N-terminal amino acid of the peptide to form a phenyl thiocarbamide derivative. So all these reagents are there basically for the identification of a sequence. So PDH amino acids can be identified by chromatography. Advanced reagent has advantage. A peptide can be sequentially degraded, liberating N-terminal amino acid one after the other, which can be identified. So this is due to the fact that peptides as a whole is not hydrolyzed but only releases PDH amino acids. Then we have another term which is sequencer. This is basically a machine to determine the sequence in a polypeptide. So we have to determine the amino acid sequence. For this we have this machine. It is based on the principle of advanced degradation. Now we have another term which is known as reverse sequencing. Now what is this reverse sequencing? It is a genetic material which ultimately determines the sequence of amino acids in a polypeptide chain. By analyzing the nucleotide sequence of a DNA that codes for the protein, it is possible to translate the nucleotide sequence into the amino acid sequence. This technique, however, fails to identify the direct sulfide bonds and the changes that occur in the amino acid after the protein is synthesized. Now we have the secondary structure. We have the secondary structure. In the secondary structure, we have two forms of a protein. We have a alpha helical structure, we have a beta sheath structure. So alpha helical is the most common spiral structure of a protein. It was come into existence in 1965 by the scientists Pauling and Corey. So in the alpha helical structure, the DNA it is tightly packed coil structure with amino acid side chains extending outward from the central axis. So alpha helix is stabilized by extensive hydrogen bonding formed between H atom attached to peptide N and O atom attached to the peptide C. The H bonds are individually weak but collectively strong enough to stabilize the helix. All the peptide bonds except the first and last in the polypeptide chain participate in the hydrogen bonding. So each turn of helix contains 3.6 amino acids and travels a distance of 0.54 nanometers. So all these are the characteristics of alpha helix. Similarly, we have a beta braided sheets which are placed one above the another. So these are also the secondary structures in which two or more peptide chains are linked together by hydrogen bond between H of NaH and carbonyl oxygen of adjacent chain. So beta braided sheets may be formed either by separate polypeptide chains or a single polypeptide chain folding back on itself. So we have the sheets which are placed one above the other, and they may be parallel as well as anti-parallel. Then after it we have tertiary structure. Tertiary structure is a three-dimensional structure of a protein. It is a compact structure with hydrophobic side chains held anterior while the hydrophilic groups are on the surface of the protein. So this type of arrangement ensures the stability of the molecule. So this is the tertiary structure, and this is your homework also, that you have to draw the primary, secondary, tertiary as well as the quaternary structure. So any one of you please write this homework and you have to type it in a group also. So we have primary, secondary, tertiary as well as quaternary structure. So you have to draw all these structures. So we have primary, secondary, tertiary and quaternary structure. So you have to draw these structures only. After it, the bonds which are responsible for the protein structure. We have covalent bonds, we have some non-covalent bonds also. So covalent bonds are there and disulfide bonds. These disulfide bond is formed by sulfur-derived groups of the cysteine residue to produce cysteine. The disulfide bonds may form in a single polypeptide chain or between different polypeptides. These bonds contribute to the structural conformation and stability of the protein. So we have different types of bonding, covalent bonding and non-covalent bonding. In the non-covalent bonding we have the hydrogen bonds. Okay? And then we have some hydrophobic bonds also. So you have to tell me the bonds also. This is again your homework. The number one homework is to draw the primary, secondary, quaternary and tertiary structure of protein. And number two, the bonds which are there. The covalent as well as non-covalent bonding or non-covalent interactions which is there in between the protein or in between the polypeptide chains. So we have electrostatic bonds, we have van der Waals forces. Like we have hydrogen bond, disulfide bond, ionic bond and hydrophobic interactions, van der Waals interaction. So we have 1, 2, 3, 4, 5 types of interactions which are there in the polypeptide chain. You have to tell me these interactions also. After it we have to determine the protein structure. Just let me check it. So whether you are getting it or not. Okay, so please any one of you, Diksha, Archana, please any one of you, Sadhan, Pooja, any one of you, please write the homework, okay, so that you will not forget it later on. Now, after it, we have the methods to determine the protein structure. For the determination of the protein structure, when we want to when we want to study the secondary or the tertiary structure, we have crystallography, we have X-ray crystallography, that is most common. NMR is also there. So NMR is nuclear magnetic resonance. It is a spectroscopy that provides structural and functional information on the atoms and groups present in the protein. So we have X-ray crystallography as well as the NMR, or you can say nuclear magnetic resonance. So these are the methods for the isolation and purification of the proteins. For the purification, for the isolation of the proteins, we are using ammonium sulfate or sodium sulfate. Ultrastratification is there, electrophoresis, isoelectric focusing, chromatography, gel filtration, all are the techniques for the separation of the proteins. These are some properties of the proteins. The solubility. The proteins form colloidal solution instead of true solution in the water. This is due to the huge size of the protein molecules. They have different molecular weight also. So their molecular weight will be range from 4,000 to 4,40,000. Shape is also varies in protein. It may be globular, it may be oval, it may be fibrous. Isoelectric pH is there when the protein exists at the dipolar stage. This is known as the isoelectric stage. Then we have some methods by which we will able to precipitate it. So by using salting out method, we will able to precipitate the protein. This is a precipitation by organic solvents. This is a precipitation of the salts by heavy metals. Precipitation is when we will get the pellet in the downside, or in the when we will get the pellet and the supernatant contains other biomolecules. Then we have denaturation. Now what is denaturation? It is defined as the disorganization of the native protein structure. Denaturation results in the loss of secondary, tertiary or quaternary structure of proteins due to cleavage of non-covalent bonds. This involves a change in the physical, chemical or the biological properties of a protein molecule. So this is denaturation, when we will able to lose the secondary, tertiary structure of the protein. This is known as denaturation. So denaturation can occur by heat, by violent shaking, by X-rays, by UV radiation. By all these things we will have the denaturation. Some chemical reagents are also there which are which leads to denaturation. Basically, we have some acids, we have alkalis, organic solvents. They are also responsible for the denaturation. Then this is again the classification of the proteins on the basis of their function, their chemical nature, their solubility, their nutritional importance. So on the functional, we have different types of proteins. According to the chemical nature, according to the nutritional part, they will divide it differently. They are classified differently. So they are divided into structural proteins, contractile proteins, enzymatic proteins, transport proteins, and regulating proteins. So these are different classes of proteins. So we have simple proteins, we have conjugated proteins, we have bright proteins also. All of you must aware of these proteins. We have albumin, globulins, butylins, protamines, stones. Similarly, we have nucleoprotein, glycoprotein, lipoprotein, phosphoprotein, chromoprotein, metalloprotein. So these are the simple proteins which are there, that they are like albumin which is there in the egg part. So I hope you all will do it by your own self. Then we have superproteins also known as albuminoids. Conjugated protein which consists of glycoprotein, lipoprotein, nucleoprotein, chromoprotein. So these are the types of the proteins according to the base on the basis of their structure only. Then we have some nutritional classification also. Nutritional classification means so nutrition classification means complete process on nutritionally rich proteins. These proteins have all essential amino acids in the required proportion. They are also called as first class proteins. Then partially incomplete proteins are there. These proteins are partially lacking one or more essential amino acids and can promote moderate growth. The example is wheat and rice protein. Then we have biologically important peptides which are there. We have glutathione, we have oxytocin, we have vasopressin, we have angiotensin, we have methionine. So all these are the biologically important peptides. So when 10 or less number of amino acids are joined together, they are known as oligopeptides. They are biologically active. So we have glutathione, we have TRH, we have oxytocin, we have vasopressin. All these are the hormones. They are important in the endocrinology part also. So that is all for this protein part. Yeah. Okay, Archana. So you have to tell me about the bonding which is there in between the protein. Then number two, you have to tell me the structure of the protein. Number three, you have to classify the proteins on the basis of their structure, on the basis of their functioning. So the third question is the classification of the protein. I know some of you have submitted the assignment, but all of you have not completed it yet. That is the thing I ask you to please complete it. Only few students are here whose assignment is complete. So I request you, and I request you to please write your name on the just before the assignment so that I will able to evaluate it. Some of you have not written the name. I request you all to please write the name also. Okay. So any one of you please write the questions in the group also so that all will be able to answer, and you have to answer these three questions. So is it clear to all of you? Please let me know. Yes, so you have submitted the structured part. It's okay, then leave the structure. Rather, you have to tell me the interaction which is there in the protein, the covalent bonding and the non-covalent bonding. Leave the structures, the primary, secondary, tertiary structure. Leave those structures. Otherwise, complete the remaining two questions. But I know many of you have not completed the primary structure part also. Only few of you have completed. So those who have completed, they don't have to do it again. Otherwise, please write these three questions in the group also, so that all will able to complete it. Okay. So is it clear to all of you? Please let me know. And I request all of you to please write the name on your assignment, so that when I will open it, I will came to know that this is assignment by Anamika, this is the assignment by Archana. Getting my point? So I request you all. Okay. So by this, our protein part is completed. Our protein part is completed. And now we have only few topics which are there, which we have to complete. We have the protein part, the structural protein structure is completed. Even we completed the properties of the proteins and their functions also. So only we have this part, the metabolism which is there. We have carbohydrate metabolism, fatty acid metabolism, muscle metabolism. Only metabolism part is left. So I hope protein part is clear to all of you. So is it clear to all of you? Please reply. So is it clear to all of you? Please reply. Hello. Please reply. Okay. So that's all for today. We will meet tomorrow with the metabolism part which is left now. So be there so that we will able to complete it. Okay. So that's all for today's session. Thank you all for being here. And if you have any doubt, you can share with me. Okay. Thank you all. Thank you all. So if you have any doubt, you can share. That's all in the protein part. Tomorrow we will start the metabolism part. And I will suggest you to complete your now focus on your review paper also. I will mail you soon, and so that you will able to complete it further. That's all for today. I will share this protein PPT with all of you so that you will be able to complete the homework also. Tissue-specific metabolism, brain metabolism, liver metabolism, muscle metabolism. Only metabolism part is left. Otherwise, we have completed the remaining things.